Hey, welcome. Our final lesson on eukaryotes, we're going to take a look at the support structures within cells. So starting off with a very fun little organelle called the lysosome. So very similar to a vesicle. In fact, they kind of are vesicles. A lysosome is just a small membrane-enclosed sac. So we see that here, the yellow phospholipid heads, the green phospholipid tails. There's our phospholipid bilayer. And it's just a sac that kind of floats around within the cell. Different from vesicles. So vesicles are going to carry, they're like transport vesicles, are going to carry proteins from one place to another. Lysosomes generally contain hydrolytic enzymes. So recall hydrolytic, what that means. Hydrolyze is the breaking apart of of just regular old macromolecules by using water to separate them into their monomer units. So these hydrolytic enzymes that are inside of a lysosome are proteins that break apart or digest macromolecules. So here's a picture of an enzyme. This one is in particular, this is sucrase, the purple blob there, and he breaks apart glucose and fructose using water. He separates glucose and fructose from sucrose. Most of the time, these hydrolytic enzymes inside of the lysosome require a very acidic condition in order to function properly. So in order to achieve these acidic conditions, lysosomes have hydrogen pumps, hydrogen ion pumps on the outside of them. That's what we have here, these green and purple dots that are constantly pumping H pluses into the interior, the lumen, so that these proteins can remain in their proper shape and digest macromolecules. So a quick aside here on acids. Acids have a high hydrogen ion, that's H plus, hydrogen ion, concentration, and we see that here. In normal old distilled water, pH is 7, that's the midpoint. We have just a few red ions, that's the H pluses, and just a few blue, that's the OH negatives. That's what makes something basic or alkaline. As you go down the pH scale, as you get closer to 0 in the pH scale, you get more and more H pluses. So notice this, we only went from 7 to 6, only one little step, and we already have, or should have, 10 times the amount of H plus concentration there. That makes it acidic. So imagine if we went all the way to 1. That would be very acidic. Lots of H pluses. So the need for that acidic condition is also a failsafe. It's actually a pretty ingenious strategy that cells have. If the lysosome membrane, for some reason, breaks apart, and some of those hydrolytic enzymes escape, we don't want them devouring the entire cell. We don't want them breaking the entire cell down. So instead, they only work in the very low pH, and if they happen to get out, if they happen to escape the lysosome, they don't have that low pH anymore. They're in the regular cell, and they can't work. So we kind of save the cell from a malfunctioning lysosome. So it's a fail-safe. Now, on the other hand, there is a very special process called apoptosis. That's programmed cell death. We'll discuss that later in units, where a cell breaks all of its lysosomes all at the same time, and there's enough increase in acidity then that the hydrolytic enzymes can function, and basically the cell commits suicide by, which is called apoptosis, by releasing all of their lysosomes all at one time and kind of breaking itself up and recycling itself back into the organism. So quick look at what lysosomes are doing inside of the whole cell. So here's kind of a picture of the whole cell. Here's the lysosomes. They're coming from the Golgi. They have those hydrolytic enzymes inside of them. And the purpose of lysosomes, so lysosomes is the whole thing, the purpose of the lysosome is that they're going to break down worn out organelles, kind of recycle those macromolecules back into the cell. Or if, in this case, the cell has some food, the lysosome is going to merge with that vesicle carrying the food, and it's going to break that down. It's going to get the important parts out of it, and then everything that's worthless kind of gets shot back out of the cell, eliminated from the cell. So we're breaking down food, digestive lysosome and getting the energy out of it, getting the macromolecules out of it, or we are devouring old worn-out organelles that might not be working and getting the energy out of them as well. Lysosomes are found in pretty much every single animal cell. Some have more than others. You see up above here, the liver cell, for example, has a ton of lysosomes. It's recycling its macromolecules, almost half of them at least, about every single week. So that's a lot of breaking down of macromolecules, recycling those parts back into the rest of the cell. So you'd expect a lot of lysosomes in a human liver cell. All other cells have them as well, maybe not quite as many, but it's a pretty common feature in eukaryotic cells. You rarely find them in plant cells, though. So animal cells tend to have them. Plant cells generally don't have lysosomes. All right, onward from lysosomes, we'll take a look at the cytoskeleton, which is very much what it sounds like. It's kind of like the skeleton of the cell. It's a network of fibers that extends throughout the cytoplasm. That's the cyto part of the cytoskeleton. These fibers are divided into three basic groups. We see them here: microfilaments, intermediate filaments, and microtubules. And that's all based up of the size of the particles that they're made up of. So microtubules, down here at the bottom, in yellow and gold, they are made up of tubulin proteins. Microfilaments, so the small ones up here on top, are made up of the protein actin. You see those as the small blue circles. And then the intermediate filaments, they're a little bit harder. We see them here in the middle, the pink. They're a little bit harder to classify based on what they're made out of, because they're made out of quite a few different proteins that all kind of twist together. But they're called intermediate filaments because they're intermediate in size. They're middle-sized, kind of between the size of a microtubule and an actin microfilament. So the cytoskeleton has a lot of jobs. Like I said, the term skeleton kind of gives you the idea of one of those jobs. It acts as kind of the framework. It keeps the cell supported, keeps the organelles where they should be, much like your bones, your skeleton, does for your body. Another very important task that the cytoskeleton, the fibers of the microskeleton do, is they act as kind of like a superhighway or a monorail for proteins and vesicles to travel on. So they go to all parts of the body, and the vesicles kind of know, hey, if I jump onto this particular microtubule, it'll take me out to the exterior of the cell. So they work as kind of a train track, set of train tracks, monorail for vesicles and proteins and other parts of the cell. The cytoskeleton is also involved in how cells move. So it's usually, when we talked about the cilia and the flagella, those are usually powered by parts of the cytoskeleton. Or the flagella is like that long tail a lot of bacteria have. Well, that long tail is powered by, is kind of a part of the cytoskeleton. Microfilaments are really important in how muscle cells work. Imagine you're climbing a rope. If you're climbing a rope, you can move up the rope by pulling on that rope, and that's kind of how microfilaments work. They kind of pull back and forth, and that's how muscles can contract and open by kind of pulling on those ropes, rope-like microfilaments. So it's important. All cells have cytoskeleton. It's not just eukaryotic cells. Prokaryotic cells also have cytoskeleton. All right, next up we have centrosome. The centrosome is an area near the nucleus where your microtubules tend to grow out of. For animals, within the centrosome, we usually find a pair of centrioles. So here's the centrosome, the whole big circle, and inside of it we have a pair of centrioles. Again, that would determine that this is probably an animal cell. We generally do not find centrioles in a plant cell. We will find the centrosome, but not the centriole. So centrioles are necessary for the cilia and flagella. Again, those are the tail-like structures. We don't know exactly what they do, but we know that if we remove them, the cilia and flagella don't work. Again, like I said, most animal cells have centrioles. Plants don't generally. but they do have a centriole, and they're very important when the cell starts to divide. They help to separate the DNA during cell division. So here's a non-dividing cell, just kind of the centrosome sitting up kind of by themselves, and here is that same cell dividing, and we can see these microfilaments coming out of the centrosome, and they're grabbing the DNA, the chromosomes, and they're going to pull them to opposite sides to kind of help separate the DNA during cell division. All right, and finally we have the cytoplasm versus the cytosol. Very similar words, but make sure you know the difference between them. The cytosol is the liquid portion of the interior of the cell. It's water-based, but it has so many salts in it, so many particles in it, that it's really going to be very, well, semi-fluid is the word I use here. So think of like a jelly. It's water-based, kind of a liquid, but has so much stuff in it, it's not really flow like water would flow. The cytoplasm, again, a very similar sounding word. The cytoplasm is not just the cytosol. It is the cytosol, but it's also the organelles. So keep in mind the difference between those two. The cytosol is just the liquid interior of the cell. The cytoplasm is the liquid and the organelles. So it's both the liquid and the organelles. So we're talking about the cytoplasm, we're talking about the mitochondria. We're talking about the liquid that surrounds the mitochondria. And neither of them includes the nucleus. So the nucleus is not a part of the cytoplasm, and the cytosol, the liquid, certainly is not. So again, all cells have both cytosol and cytoplasm, but as you can imagine, the cytoplasm of a prokaryotic cell is going to be a lot different. It does not have nearly as many organelles. In fact, it shouldn't have any organelles, except perhaps ribosomes. So the cytoplasm of a prokaryote is going to be a lot simpler. All right, I believe that does it. That is the end of our cells unit. That's the last bit. So go study up on cells, you know all their cell parts, and we're moving on to another unit.